Hey everyone, welcome to this masterclass, all about the program code that runs us, none other than DNA. In this masterclass, you will learn all about DNA step by step. We will take you through all from the basic level to the advanced level. So without further ado, let's get started. What is DNA? Every living thing has DNA, from plants to animals to bacteria, even to viruses which were once considered non-living. And most importantly, we humans have it. DNA stands for deoxyribonucleic acid, and although every living thing has DNA, its very composition differentiates you from others. The interesting fact is that humans' DNA is 99% the same, therefore only 1% of DNA that makes you unique. Does this mean twins don't have the exact same DNA? No, they don't, unless they are monozygotic or identical twins. So now, questions arise. Why every living thing has DNA? What is so important in it? And what does it do? DNA has a recipe for synthesizing proteins. DNA synthesizes RNA or ribonucleic acid by a process called transcription. This is further converted into proteins by a process termed as translation. The explanation of DNA conversion into RNA and finally RNA into proteins is called central dogma. DNA contains hereditary information that is passed on to you from your parents. If you and your father have the same color of eyes or your hair are as silky as your mother, this is because of the DNA that you inherited from your parents. Even many diseases are passed on from generation to generation because of DNA. Where does DNA reside in your body? Your body is made up of billions and trillions of cells, and almost all cells have DNA in their nucleus. Can you tell us which cell may not contain DNA? Now, let's talk about the structure of DNA. Think of DNA structure as a ladder whose rungs are made up of different bases and sides are sugar phosphate. Each composition of this nitrogenous base, pentose sugar, and phosphate group is called nucleotide. The nitrogenous bases are of 4 different types: adenine, guanine, cytosine, and thymine. Adenine will always bind with thymine and cytosine with guanine. This type of bonding is hydrogen bonding. Phosphate and sugar are linked together to make the backbone, and complementary bases are attached to it. Due to the bond angle of the sugar-phosphate molecules, linkage will eventually form a double helix structure. That's all the basics of DNA. There's a lot more to know coming right up.
So you have now completed the basic level of this masterclass. Now let's level up. The structure of the DNA molecule was discovered in the early years of 1950s. Note that the structure was discovered, not the molecule. The discovery of DNA molecule predates to the 18th century. But DNA was ignored for a long time, and it was proteins that grabbed a lot of attention. However, post-Griffith's transformation experiment, the other 2 notable works in the timeline helped shift the focus towards DNA. So around the early years of 1950s, an English chemist named Rosalind Franklin started working on DNA molecules. She isolated the DNA crystals and shone X-rays on them. As a result, she obtained fine images of the structure of DNA molecules. According to sources, the best one looked somewhat like this. Around the same time, Francis Crick and James Watson, 2 scientists in the field of genetics, worked on the structure of DNA and reached a similar conclusion like Franklin did. However, according to sources, they were fortunate to receive better and earlier recognition compared to Franklin. They even shared a Nobel Prize with Maurice Wilkins for their discovery of the double helical structure of DNA. Let's understand how exactly this double helical structure looks. We may have come across the structure of DNA somewhere. Is this how it really looks? Yes. According to Franklin, Watson and Crick, the DNA is made up of 3 major components: One is the pentose sugar molecule called deoxyribose. How is this obtained? It is obtained by removing one oxygen atom from a ribose molecule. Thus the name deoxyribonucleic acid. The second component is a phosphate group. This helps bind the two deoxyribose molecules together. The third and fifth carbon atoms of the adjacent deoxyribose molecules are bound by phosphate groups. Lastly, the nitrogenous bases form the core of the double helix. These are four different molecules that bind in pairs. Two of them are called purines and other two are called pyrimidines. Adenine and guanine are the two purines which bond with the pyrimidines thymine and cytosine. And can any purines bind with any pyrimidine? No. It is a thumb rule that adenine always binds with thymine while guanine binds with cytosine only. These molecules are bound to each other with the help of hydrogen bonds. Adenine binds with thymine with the help of two hydrogen bonds. Guanine and cytosine, however, bind with three hydrogen bonds. Did you know that one unit consisting of a deoxyribose molecule, one nitrogenous bases, and a phosphate group is called a nucleotide? And what if the phosphate group is removed from this nucleotide unit? It is called a nucleoside then. Nora wants to know if there is any way of measuring a DNA molecule. What about its dimensions? The dimensions of DNA are very simple and easy to remember. The diameter of the double helix is around 2 nanometers. The double helical strand has one major and one minor groove that makes up 110. The length of 110 is around 3.4 nanometers. And there are around 10 base pairs in 110. Now can you help me with the distance between the two adjacent base pairs? It's simple. The distance is 0.34 nanometers. It's simply 3.4 divided by 10. This is how the molecules are arranged. So in this double helix, this portion is the backbone which is made up of the deoxyribose molecules and the phosphate groups. Whereas this portion that forms the core is made up of the nitrogenous bases.
Now you have completed the intermediate level. Now let's step up a notch. This is DNA or deoxyribonucleic acid. During this video, we will look at different representation of this DNA molecule to better view certain details, but all the view represent the same structure. Inside the cell, you will mostly commonly find a double-stranded DNA in which two strands intertwine together to form a DNA double helix. The most common form of DNA double helix is what we discuss here is called B-form DNA. Now let's move to more simplified representation of this DNA structure. We can unwind the double helix like this, so that we can see so many chemical structures inside it. Each strand is a polynucleotide, means that the strand is made up of many individual units called nucleotides. A nucleotide has three components: a 5-carbon sugar, a phosphate group, and one of 4 possible nitrogenous bases: adenine, guanine, thymine, and cytosine. The nitrogenous base is always attached at the 1' carbon of the sugar. If we count on there, we can see that a phosphate group is between the 5' carbon of one sugar and 3' carbon of nearby sugar. The sugar is called deoxyribose because it is missing a hydroxyl group at the 2' carbon which is present in the ribose. Because of these nucleotides in DNA, deoxyribonucleic acid are called deoxynucleotides. Nucleotides attached at the each end of the DNA strand by phosphodiester bonds. The phosphate group of one nucleotide binds to 3' oxygen of the nearby nucleotide. Thus we can see that the sugars and phosphate group make up the DNA backbone. The carbon numbering is key to describe the directionality of DNA strand 5' to 3'. Looking within the sugar, there is an intrinsic orientation. On the top strand, you can see 5' carbon of sugar and left and 3' carbon is on the right. The opposite is true on the bottom strand. Reading left to right, that makes the top orientation will be 5' to 3' and the bottom strand orientation will be 3' to 5'. These strands are also sometimes called Watson and Crick. Keep in mind that we are only just simplified this representation by unwinding them to see more information about it. Although the nucleotides come together through covalent bonds in the backbone by the two double-stranded DNA interact together between the bases. Each bases forms multiple hydrogen bonds with its complementary bases on the opposite strand. Bound together by hydrogen bond and each units called are base pairs. The hydrogen bonding contributes the specificity of the base pairing. Thymine pairs to adenine with 2 hydrogen bonds and cytosine pairs to guanine through 3 hydrogen bonds. Thymine and cytosine are called pyrimidines, and adenine and guanine are called purines. The geometry of AT or TA or GC and CG base pairs have same geometry and they can form same symmetry and base stacking. This is mostly has to do with the distance between these backbones. Other base pairs like GT, for example, it cannot form same geometry and cannot form strong hydrogen bonding. The double helix structure of DNA is highly irregular. Each turn of the Ely measures base pairs of will be 10. In addition to hydrogen bonding between the bases, the stacking of the bases also stabilizes the double helix. The pi-pi bond interaction form when the aromatic rings of the base attack the nest of the other and share the electron probabilities. The regularity of helical structure forms the two repeating and alternating spaces called major and minor grooves. These grooves act as base pairs recognition and binding sites for proteins. This major groove contains base pair specific information, while the minor groove is largely base pairs non-specific. This is because of the pattern of hydrogen and donors of protein can interact with the grooves. In the way, the DNA can be acted upon in the specific sequence and non-specific sequence, and it will be carried out for that designated task. This is DNA double helix, and you have now learned the structural features by the influence and its function. We hope you have enjoyed and exploring this amazing molecule with us.